Chapter 20 is going to look at DNA tools. So these are the technologies that we use to manipulate DNA. And then biotechnology. So biotechnology is how we take these tools and leverage them for human consumption. There are three main technologies that we're going to talk about in this sequence. DNA sequencing. So this is the first step in DNA technology, is what does the DNA actually say? Restriction enzymes, so these are going to be used to cut DNA, to insert DNA into specific sequences. And then polymerase chain reaction. So restriction enzymes and PCR, polymerase chain reaction, are both used for DNA cloning. So a lot of the times when we work with DNA is such a small molecule that we need to make copies of it if we're trying to work with a specific gene. So we want to make millions and millions of copies of it, and both of those will give us a way to do that. Biotechnology is slightly different. So biotechnology is the manipulation of organisms or their components, such as DNA, to make useful products. So this is talking about making genetically modified organisms and making things that humans are going to buy and consume. So first is DNA sequencing. There have been several implementations of DNA sequencing. The first was by a scientist named Sanger, who had a very slow, tedious method of doing DNA sequencing by looking at individual nucleotides using radioactive labels. In the generations after Sanger, we implemented what's called next generation sequencing techniques, which took the same basic idea but used computers to compile DNA fragments that are copied. So what we can see in the results down here is this is a fragment of DNA, and we use radioactive labeled nucleic acids to pair with these fragments. And we put radioactive labels on these nucleic acids. So whenever a nucleic acid is found, it gets labeled as radioactive. So you can see these results. Anytime the bar reaches up to 1, that's going to indicate 1 nucleic acid. And you can see that that fluoresces is the color T. So this would indicate that there was a thymine here. The next one, again, we're at 1, and it's yellow for C. So that would indicate that this is a cytosine. When there's 2, so you can see that this green bar comes all the way up to 2, that's an A, so that's going to indicate that there's 2 adenines in that spot. So you can see that this one has been translated out for you. There's 2 thymines there. This yellow is a cytosine right there. Then another thymine. The next one here is orange, that's the guanine. And so doing this by hand is extremely tedious. Instead, this fragment of DNA would be fed into a computer, and then the computer would read this results and give you the DNA sequence. And then all of the different fragments that had been used would be compiled in the computer to give you an entire sequence of the entire genome. Next generation sequencing like this is the most common, and it is still used today, especially by scientists who are sequencing large genomes or lots of DNA. It's a very computer heavy process and requires a lot of kind of processing power. Third generation sequencing is kind of the next step. Most often, this is going to be new techniques that are faster, less expensive. These are typically either used in conjunction with next generation sequencing or sometimes replace next generation sequencing altogether. One example of this is the nanopore sequencing. So what this does is it runs a single strand of DNA, so you can see here's a DNA strand, through a tiny, tiny small pore in a membrane. So here's the nanopore. And what happens is as it goes through here, it disrupts the ionic current. And each current change is respective of one of the 4 nucleic acids. So based on what the current says, we can tell whether it's a cytosine, adenine, thymine, or guanine. These are really exciting. These are absolutely tiny. So here you can see the pipette with the DNA, inserting it into the nanopore. So you can run an entire DNA strand on something the size of a thumb drive and then plug it into a laptop and read that sequence even in the field. We have sequenced millions of genomes. So by the time the human genome was done, even a few years later, we had started sequencing many, many other organisms. There's a free catalog online called GenBank. So if you go to this website, you can find free sequences that people have posted to this online resource. Most of the time when we want to study a gene or make something with a gene, we can't do it with a single piece of DNA. A single piece of DNA isn't very much to work with. So a lot of the times we need to do processes that are going to allow us to clone the DNA and make many, many copies. A plasmid is something that we find in a bacterial cell. So this is some kind of prokaryotic bacterial cell. This is the bacterial DNA, so this is the chromosome. And this is a 1 chromosome, and it's a circle. You can see it's all contained here. Plasmids are extra small pieces of DNA that are not part of the main bacterial chromosome. So over here you can see this cell has 1, 2, 3 plasmids, and they're just teeny tiny circles of double-stranded DNA. A lot of times plasmids in bacteria are going to convey resistance to antibiotics, but plasmids have proven to be something that DNA researchers can work with. So what a researcher can do is we can extract a plasmid and we can cut it using something called restriction enzymes, and it'll cut in a specific spot, and we can insert a gene into the plasmid. So here you can see they have inserted the gene for insulin. Insulin is the hormone that's going to help diabetics to regulate their blood sugar. And so people who are diabetic either don't produce enough insulin or they have resistance to insulin. So either way, they have to take insulin to help their bodies regulate blood sugar. So what this is going to do is we're going to put the gene for insulin into this plasmid, stick this plasmid back into a bacteria, and then let the bacteria reproduce. If you remember, bacteria reproduce very quickly. So all they have to do is divide, and then you've got 2 bacterial cells, and each one will have a copy of this plasmid. So we can let a bacterial colony reproduce for a day, and we can make millions and millions and millions of copies of this gene inside of those bacterial cells that we can then later harvest. So here's how restriction enzymes work. Restriction enzymes are molecules inside of bacteria that cut DNA in specific sequences. So these are a mechanism for bacteria to use as a defense against viruses. So if a bacteria gets infected by viral DNA, restriction enzymes can often cut up the viral DNA and allow it to prevent it from being infected by that virus. So we have discovered restriction enzymes inside of bacteria. So now what we do is we actually take those enzymes out and we harvest them and use them for ourselves. Bacterial restriction enzymes are going to cut at specific DNA sequences called restriction sites. So this specific enzyme that we're going to use in here is going to cut at the sequence GAATTC. And you'll notice that on both strands it says GAATTC from 5' to 3'. And so these restriction sites are always going to cut at a specific spot. So this one is going to cut between G and A. It's going to leave this DNA cut into 2 pieces. And those 2 pieces results in restriction fragments. So this is a fragment and this is a fragment. Because of the way that restrictions enzymes cut, they leave a single-ended strand called a sticky end. So right here you can see the sticky end. So the sticky end reads AATT on this side, AATT on this side. So those are the sticky ends. If we then take our insulin gene and we cut it with the same restriction enzyme, it will end up with the same sticky ends, AATT, AATT. So now our restriction fragments and our target gene actually have the same sequences to match back up. So if I put these together into a container, they will align back together. And so we will get fragments that will align with the target gene, and you can see how they match back up. What we need to do then is add DNA ligase. That's the one that's going to bind fragments back together. And here we go. After the ligase, we have double-stranded DNA. It has been cut, and our target gene has been inserted. So this is called a recombinant DNA plasmid or DNA molecule. Recombined just means that it's got DNA from 2 different organisms. So now this recombinant DNA, we're going to need to check it. So we check the recombinant plasmid by running it on something called a gel electrophoresis. So a gel electrophoresis is here. There are several wells right here that are places you can insert DNA. And this is a porous gel. DNA has a slightly negative charge because of all of the phosphates in the background. So when we run a power source through it, where we have a negative over here and a positive pole over here, the DNA will move along the gel like this. The larger molecules don't move as far because they're really big and it's kind of moving through a gel. So molecules that are closer to the negative end are going to typically be larger molecules of DNA. The ones that get to move further down here are going to be smaller pieces of DNA. Those are going to be able to move through the gel much faster. And so what we end up seeing is a series of bands of different fragments of DNA that spread out. So when we are making a recombinant plasmid, we know what kind of bands we should be looking for, and we can run the gel and check it to make sure that we actually made the plasmid we were looking for. Gel electrophoresis is also used in all DNA technology. So if a scientist receives a sample of DNA that they're going to use as a study for their research, they check it on a gel first to make sure that it's the correct fragments and size and DNA that they were looking to use. Another way to make copies of DNA, and this is used to make specifically copies of small fragments of a targeted segment. So if we know that there's a specific gene that we want to make copies of, then we can use PCR or polymerase chain reaction to make many, many copies of this very, very quickly. So this goes through a 3-step process. So we start with a bunch of items that we need inside of a test tube. So we have our target sequence, which is the target gene that we want to make a copy of. We're going to have some nucleic acids. So these are nucleotrisophosphates. So those are going to be the adenines, the guanines, the thymines, the cytosines. Those are going to be the new nucleic acids that we're going to be adding. We've got some primers because we know we're going to need primers for polymerase to work. These primers are going to specifically pair with the target sequence that we want to work with. So they're going to specifically target the sequence that we want to copy. And then we have a DNA polymerase. This DNA polymerase is a special DNA polymerase. It was found in bacteria that live in hot sulfur springs. It is called Taq polymerase. It is not the DNA polymerase that's found in humans. This is a heat-stable DNA polymerase. So this is going to be able to function at a very high temperature, unlike the DNA polymerase that you find in our body. So the first step is to open the double helix. So we do this through denaturation. So this is where we do heating. So at this first step, you would heat up the contents to separate all of the DNA strands. What this does is it breaks all of the hydrogen bonds between the two DNA strands, and it's going to allow them to separate. The second step is annealing, so this is cooling. And while it cools, the primers are going to go ahead and bind to their complementary sections. And then the last step is replication. So this is going to allow for that Taq polymerase, so here's my DNA polymerase there, to add the nucleotides into the complementary base pairs. So it'll work from the primer, 5' to 3', just the same as we do in DNA replication, to add these new DNA nucleotides into this. So at the end of the first cycle, we started with 1 double-stranded DNA, and we ended with 2. Every cycle will double the number of copies. So in the second cycle, we start with 2 and we end up with 4. So 2 to the 2 equals 4. By the end of the third cycle, we started with 4 and we end up with 8. 2 to the 3 is equal to 8. So every time we do these cycles, we can get 2 to the n number of copies. So this is going to be very quick. It doesn't take too long to do a single cycle of PCR. In a couple of hours, you can do 40 cycles. So let's say we're on n equals the 40th cycle. So 2 to 40 is a really huge number. It's over 1 trillion copies of DNA. So it's not just double, it's doubling every time, and doubling gets very big very fast. So PCR is a really efficient way of making lots of copies of a very specific small piece of DNA. And this is called DNA amplification, or making lots of copies of. So once we know what the DNA says, which we can make through DNA sequencing, and we can make many, many copies of DNA, now we need tools for understanding what the DNA actually does. So DNA expression is through proteins, and one of the best ways to figure out which genes are being expressed is to look for the messenger RNAs that indicate that it has been transcribed and is in the process of being translated into proteins. And then we can look at the different types of cells that are expressing the different types of genes and figure out what those genes do. So one type of viewing this is called nucleic acid probes, and this will look at a specific gene at a time. So what you do is you soak the cells in a radioactive tag, and then eventually look at which of the cells are fluorescing or expressing that radioactive tag. So here in this image you can see a Drosophila, which is a fruit fly embryo, and it has been soaked in 5 different probes, so you can see the 5 different colors. And so along some of these cells, they're fluorescing bright yellow or blue, green, orange, or purple based on which genes are being expressed in those cells. Sometimes we want to know more than just is the gene being expressed, but we want to know how much of it has been expressed or how much of it is there. So one thing we can use is something called reverse transcriptase, PCR, to look at how much mRNA is inside of a cell, and then use that as a way to figure out how much mRNA was present, not just whether it was present or not, but we can actually figure out relatively if there was more or less. So this process is going to kind of start with RNA, go back to DNA, and then use PCR to make copies of that DNA. So we start with the messenger RNA. This is the expression of the gene. And then we use a primer to build a complementary DNA strand. And then we break down the messenger RNA. And we use the DNA polymerase to build the template DNA strand as well. This DNA is called complementary DNA because we're going from messenger RNA to DNA, so we're not going to have any introns available. If there were introns in the gene originally, they would have been removed during RNA processing when we made the messenger RNA. So when we go from messenger RNA back to DNA, this is going to be called complementary DNA or cDNA because it's slightly different than the original gene. However, a computer will be able to pick up on this difference and match it to the correct gene. Then what we do is we take the cDNA and we run it through PCR, principal component polymerase chain reaction. We can tell how much there is because it doubles every time we do PCR. So if we have a certain threshold that we're trying to hit, we can just figure out how many turns or how many rounds of PCR it took to get to that specific threshold. The longer it takes, the less DNA was available in the beginning, meaning that that cell wasn't expressing that much of that gene. This is actually how the COVID-19 test is done. So COVID is a coronavirus, which is an RNA virus. So what they do when you get a COVID test is they swab your throat, and what they're doing is trying to collect viral RNA on that swab. They then run the viral RNA through reverse transcriptase, which is going to use this enzyme called reverse transcriptase to turn it into cDNA. Then we run PCR on the cDNA, and depending on how many rounds it takes to hit a certain threshold amount of COVID virus, you can say whether the viral load of a patient was very high and is more likely to have a severe reaction, or it was very low if it takes more rounds of PCR and is more likely to have a moderate reaction to the virus. Those are all been methods of finding out if a single gene is being expressed. A lot of times we want to see if multiple genes are interacting. This is often used for finding genes that are associated with specific disorders. So a first tissue might be a normal tissue, and then the second tissue might be a tissue with some kind of disease. So for example, it could be a cancerous tissue. So what we want to do is we want to look at many genes across these tissues and see which genes are being expressed in the disease tissue, but not the normal tissue, to see which genes might be affecting that disease. So what we would do is run the genes from these two different types of tissues across something called a DNA microarray assay. So this would take the genes from the entire genome and cut them up into very small sections, and then run them across this microarray, which is a glass tray with very small fluorescent pits in it. And what we would see is if a spot fluoresced orange, in this case, that would indicate that that is a gene that is being expressed only in the normal tissue. And then if a specific gene fluoresced green, in this case, it would indicate that that gene is being expressed only in the second tissue or the disease tissue. So that might be a gene that we would want to look at in determining the prevalence of the cancer or something like that. An area that fluoresced bright yellow, which is kind of a mix of the orange and green, would indicate that that's a gene that's being expressed in both tissues. So maybe it's got something to do with the disease, but maybe not. And then something that is black and doesn't fluoresce is going to be a gene that's not being expressed in either of those tissues. So this procedure can be used to look at the entire genome of a tissue in two different situations to figure out which genes are on in one situation and which genes are on in the other situation. RNA-seq is another example of looking at kind of an entire genome for tissues in different stages. So this is often used to look at a tissue of maybe an embryonic stage, so during development, versus a adult stage. So what we would do is take the mRNAs from that tissue, cut them up into small fragments, and then use reverse transcription again, to go from RNA to DNA. So you can see we go from single-stranded RNA to double-stranded DNA. Those DNA fragments would then be sequenced, and then a computer would take all of those DNA fragments and overlay them to get the entire genome sequence of a specific tissue. So then you can map those specific sequences onto the genome to figure out which genes were being expressed in RNAs and which ones were not being expressed in those specific cells. So a lot of times this just tells us which genes are on and which genes are off. So which ones are being made into proteins because they have mRNA present, or which ones are not. But what's another hard question is determining the function of a gene specifically, without knowing necessarily what its protein is or what it's doing at any one time. So we have a very cool tool called CRISPR-Cas9. And this is a combination of a protein called the Cas9 protein and a guide RNA. And this is going to allow us to target a very specific sequence. So we can make the guide RNA have a complementary sequence to whichever gene we're interested in studying. One of the best ways to study what a gene does is called a knockout. So what you want to do is you want to go in and deactivate that gene and then see what happens when it's broken. Because oftentimes when it's broken, you can figure out the difference between when it's fixed and when it's broken and figure out what that gene actually does. So this is the CRISPR-Cas9 system, is a guide RNA and this Cas9 protein. So this is inserted into the nucleus. And the guide RNA will take the Cas9 right to the target gene. So here you can see the target gene is going to complementary base pair with this guide RNA. This CRISPR-Cas9 acts like a pair of molecular scissors. So right here you can see the Cas9 active sites. And what that's going to do is it's going to cut right through both of these double-stranded DNAs. And that's going to result in the target gene being completely cut. So after we cut the gene, we can actually edit it. So using this, we can edit the gene. So sometimes we do what are called knockout studies. And so once the gene is cut, we will insert some random nucleotides, which will make it not function anymore, or it is knocked out. And then we can look at the effect on the cell. So we can go back to the overall cell and see what changed. So when we take a functional gene and we turn it off by knocking it out, how does that affect the cell? And if we see an effect, then we can attribute that effect to the specific gene of interest. And now we know what the gene actually does. The really cool thing about CRISPR-Cas9 is if the target gene was actually a mutation, so maybe instead of doing a knockout study, what we were trying to do was repair a cell, we can actually go in and take out the mutated section and put in a normal wild type normal nucleotides. So you can take something that has been mutated or is causing disease and actually replace it with the correct nucleotides in order to repair that DNA. So this is a potential use for gene therapy or editing genes in order to go from maybe a mutated disease like sickle cell or cystic fibrosis and fix it to make it so that it works properly. So there's a couple other methods for studying gene function. One of them is called RNA interference. Expression of a gene, so this is kind of like a knockout study here too, where we're going to stop the expression of the gene by silencing it. In order to do this, what we're going to do is insert RNA-I, which is an interfering RNA, and that's either going to break down the messenger RNA or completely block translation. So what this is going to do is it's going to allow a gene to be transcribed into RNA, but it's going to stop it from being translated into a protein. So effectively, the gene is turned off, and we don't actually see the result of the gene. And so once the gene is knocked out, again, we can study it and see what happened. Again, we can study it and see what was the effect in the cell, and then we can attribute that effect to the specific gene. Sometimes we want to study genetic markers at a larger level. So oftentimes we'll study populations of people and look at their genetics that have maybe a specific disease. And so what we'll find is something called a single nucleotide polymorphism, or a SNP. And it's pronounced snip, like you would snip something with scissors. And so sometimes in a population of people with a disease, we can find a specific single nucleotide polymorphism that is different than the people who all have the normal allele. And if an entire population of somebody with a specific disease has the same SNP, then we can look for the SNP in people who might potentially have the disease. So then we can associate this SNP with a disease, and then later on when we do genetic analysis, so companies like Ancestry.com that allow you to do a cheek swab and look for diseases would look for the SNP instead of looking for the entire genome to find the gene associated with the disease. So this is more of a large-scale analysis on the order of entire populations who do or do not have a disease. One area that is a large area of interest of study in biotechnology is the idea of cloning, or taking one cell and copying it into another cell. And this is done mostly because it is going to be able to produce stem cells. So a stem cell is an unspecialized cell. So this is a cell that has not yet decided what it is going to be in its adult form. And there are several different kind of levels or types of stem cells. So what you can see in this image is this is a fertilized egg. This is embryology all the way to a full embryo. A full embryo is going to happen at about 12 weeks during pregnancy, once all of the organs have started to kind of decide what they're going to be. So during the early stages of blastula, and this is going to happen just a few days after fertilization, the developing embryo isn't actually an embryo yet. What it is it's a group of embryonic stem cells, in this case ES, embryonic stem cells. Embryonic stem cells are called pluripotent. Potent, think about the word potential. And then pluri means everything, so all potential. So these embryonic stem cells have the ability to become endoderm cells, which are the internal tissue layer, mesoderm cells, which are our middle tissue layer, and then ectodermal cells, which are our outer tissue layer. So these are all of our tissue layers. So stem cells that are embryonic stem cells have the ability to differentiate into all of the different cells in your body, which is necessary during embryology because you eventually will need all of the different cells in your body. Adults do have stem cells. So adult stem cells are usually multi, omni, or unipotent. So adult stem cells would be considered like hemopoietic stem cells. So these would be stem cells in your red bone marrow that could become different types of blood, white blood cells or red blood cells, but hema means blood. They only become blood cells. So the adult stem cells can generate sometimes multiple different types of cells. And this is usually to replace non-reproducing cells. So if you think about red blood cells, they don't have a nucleus. They are not able to reproduce on their own. Red blood cells can usually live for about a month, maybe 2 in your body, and then they die. So they have to constantly be replaced by these blood stem cells or hematopoietic stem cells. Your intestinal lining is also always being replaced, and it's because you have intestinal stem cells that are going to continue to make intestinal lining cells. Just like your skin is always being replaced, the skin in the bottom layer of your epidermis consists of epidermal stem cells that can then continue to go through differentiation to make new skin cells. So those are examples of adult stem cells. They can make certain types of cells, but they're not pluripotent. They can't make every single type of cell. You can't take an intestinal stem cell and grow a whole human. You can take an embryonic stem cell and grow a whole human. Plants don't have the same issue. Plants actually can mature into differentiated cells, and then those cells can actually be de-differentiated or turned back into stem cells. These types of cells are called totipotent. Potent, again, meaning potential, toti meaning all. So it has the potential to become an entire organism. So total is entire organism. So this is often used in agriculture for plant cloning. This is pretty much the only feasible way to sell orchids. So once you have a specific type of orchid, you don't want to let it go through sexual reproduction and then kind of risk getting the next generation of not this specific orchid. So a lot of times what they do is they will take the specific strain of orchid we want, allow those mature stem cells to form into something called a callus, grow them on a bacteria, and then eventually generate an entire plant from specific cells. That way you end up with exact clones of your orchid, and you always have a reliable product that you're going to be able to sell to customers. We have, of course, experimented with cloning entire animals. We typically are going to see this done through a process called nuclear transplantation. So what this is going to do is it's going to take a nucleus of an unfertilized egg. So here's a frog egg that has not been fertilized, and it is going to be enucleated or remove of the nucleus. So you can do this with UV radiation, and that is going to destroy the DNA in the nucleus, and then you end up with an egg that has everything we need to produce a tadpole, except the instructions, the chromosomes. So what we would want to do is take a donor nucleus, and this donor nucleus would come from a different cell. So sometimes we would take it from a frog embryo, so this would be a less differentiated cell. So this would be taking a kind of a younger nucleus. Or we could take a nucleus from a fully formed frog tadpole. This would be kind of an older nucleus. And then we stick it inside of this unenucleated cell. Typically we find that the younger donors are going to be more successful. Most of these are going to be able to develop into a tadpole. The older the donor, so if we take a fully differentiated cell from a tadpole's intestine, stick it in a tadpole egg, it might develop into a blastula here, but a lot of the times they're going to stop developing and not actually turn into a full-on tadpole. So this actually supports the idea that the genes that are being expressed at different stages are different. So because the frog embryo donor was more similar in stage to the egg, it was more successful at developing into a tadpole. Whereas because this older frog tadpole was an adult stage compared to this egg, it was not as successful because it didn't have the right genes on or off during this development phase. And then past frogs, we have, of course, cloned a few mammals. So the first successful cloning of a mammal from an adult differentiated mammary cell. So what we did is the donor cell was a mammary cell, so that's going to come from a mammary gland on a lamb. And in 1997, we were able to successfully clone all the way till birth a lamb that was called Dolly. And so Dolly would be an identical clone to the mother who donated the mammary cell. However, Dolly had a lot of issues. She only lived until 6 years old when she had to be euthanized because of complications. So it's possible that by using an adult donor, there are certain developments that kind of don't go correctly during development, and so Dolly wasn't able to survive past 6 years old. That there's speculation that clones of adult animals are not actually as healthy as those of the actual adults. The first cloned calico cat was named Carbon Copy. And you can see here the mother and the clone. And what you'll notice is even though these two are identical genetically, they have different patterns of orange and black. And that's because of the X inactivation. So after the DNA has already been decided on, even though these individuals are identical cloned cats, the fur patterns are different because some fur patches in one cat are orange and the other cat had a different X chromosome become inactivated for fur color. So even identical individuals can have changes that are reflected in their physiology. So things that we've learned from cloning mammals is that most of these nuclear transplantation studies, only a small percent of the embryos actually develop to normal birth. And many of the cloned animals exhibit defects. So like Dolly, who died early because of defects and then eventually had to be euthanized because of complications from those. There are also epigenetic changes. So epigenetic changes are things other than the DNA sequence. So this is histone acetylation, DNA methylation, things that get passed down from one parent to the next that might be changed during development. And so these might be specifically required for early stages of development, and the clones don't get the proper epigenetic methylations because they're getting DNA from an adult rather than going through embryolysis from a zygote. And so we think that this might have something to do with some of the kind of fragile clones or diseases that we see in some of these cloned organisms. So again, DNA technology is the tools that we use for manipulating DNA, like sequencing and cloning. Biotechnology is taking the manipulation of DNA and organisms and their components in order to make useful products for humans. This could include medicine, practical things like genetic profiles for court cases, environmental cleanup, agriculture, genetically modified organisms, all sorts of stuff that we use either in certain contexts or even in our day-to-day lives. So we use DNA technology for treatment and diagnosis of diseases. This is a really big one. The identification of specific genes and specific mutations that play roles in genetic diseases has been a huge boon for us because now we can figure out who has a disease based on their genetics or is more likely to get a disease. And we can use that to treat those diseases as well, to develop therapies. RT-PCR, for example, is really good at detecting viral amounts. We are using this now for the COVID-19 test. So even a small amount of HIV in the blood can be detected with RT-PCR and it can tell us how much our HIV is there. So we can use this for early detection of HIV and potential therapeutic drugs to help prevent HIV from becoming full-blown AIDS. And so somebody with HIV can now live a long, happy life without a death sentence looming over their head in their young 30s to 40s. PCR, so polymerase chain reaction, has been used to diagnose hundreds of diseases. So once we know certain genes or mutations that are associated with some diseases that are specifically very deadly or late onset, things like Huntington's disease, where we can look for a specific allele, we can use PCR to look at the genome and determine definitively whether somebody does or does not have Huntington's disease. And then if somebody does have a disease like Duchenne's muscular dystrophy or Huntington's, where these are kind of degenerative disorders, there are preventative therapies that that person might be able to go through to help prolong their healthy life. We also use diagnosis for personal genome analysis. So we know of many SNPs, or single nucleotide polymorphisms, that are associated with specific diseases. Things like heart disease or certain types of cancers have increased risks if you have some of these certain genes. So we can run genome analysis, and we can do this now for $100. You can send a spit swab to places like Ancestry.com or 23andMe, these direct-to-consumer genome analysis companies, and they will give you an entire profile of your genome analysis. Are you at an increased risk for heart disease, cancer, all sorts of different things based on some things like SNPs and your actual genes. Gene therapy is the idea of taking somebody who is inflicted with a mutation, or they have a defective gene for some kind of issue. So for example, this could be cystic fibrosis, which is going to cause huge issues with breathing because it builds up in fluid in the lungs, and it causes people with a cystic fibrosis disease to not be able to breathe effectively. One current treatment for people with cystic fibrosis is to get a lung transplant. Wouldn't it be way cooler if we could just insert the normal allele into the lungs to replace the defective allele and allow the tissues that are being affected, so in the case of cystic fibrosis, maybe that would just be the lungs, to be fixed on their own by changing the DNA. So typically the way that we would do gene therapy is by using viruses to insert the gene into body cells. So here you would have the normal gene that we want to put in, and we insert that into a virus. We allow the virus to infect those cells. Obviously, we would knock out the actual viral DNA or make it so that it's a non-infectious virus. All it's going to do is insert the gene that we want. Bone marrow is a really good candidate for inserting viral genes for gene therapy because bone marrow has those multipotent stem cells that can develop into other types of cells. And so that's going to be a cell that's going to continue to reproduce. This type of gene therapy was done in France in 2000 with 10 young children with SCID, which is severe combined immunodeficiency disease. People with SCID basically have no immune system and they die very young. So of these 10, 9 of them were able to show improvement after 2 years, but eventually 3 of them developed leukemia and 1 of the children with leukemia did die. However, that means that 8 out of 10 were able to survive and live longer than they would have if they hadn't been treated with this gene therapy. One of the ideas is that these viruses, there's no way to tell the virus where to put the gene. So if the virus puts the correct gene into an area that is near the proliferation of blood cells, what it could do is cause the body to produce too many white blood cells and result in leukemia. It would be way better if we could specifically tell the virus where to put the correct gene. So the CRISPR-Cas9 system is actually a really cool idea. We haven't done this much in humans. There's one researcher who reports that he has used CRISPR-Cas9 in humans, but for the most part, this is still being done in rats for now. But the idea behind this would be that you can use the guide RNA to tell the CRISPR to go specifically to the gene for SCID or cystic fibrosis or sickle cell anemia or whichever type of allele we're trying to fix. Then what you could do is you could cut the defective gene and put in the correct functional gene so you could repair it. In 2018, we saw a promising study doing this with sickle cell disease in mice. And so what they did is they took sickle cell from patients who are adults who had sickle cell. They edited them and then injected the edited cells into the bone marrow in mice. And what they found is that in 20 to 40% of the injected cells, the gene remained correct after about 20 weeks. So this means that it would have improved that person's outcome because they would have more normal blood cells than sickled cells. In 2018, a Chinese researcher also reported that he used the CRISPR-Cas9 to edit the genes in embryos. So this was human embryos for the CCR5 gene. This is a co-receptor for HIV. If the CCR5 gene is broken, that person cannot receive the HIV virus. So these were done in a set of twins and then another individual whose father was positive for HIV. And so they were attempting to prevent the children from contracting HIV. This was very controversial. I don't believe he had permission from the government agencies in order to do this. So CRISPR-Cas9 kind of presents this really promising potential molecular scissors to go in and snip out a defective gene and then replace it with a different or correct or normal gene. So is this gene therapy ethical? Should we be doing this? I think most people will agree that, making people who are diseased not diseased, maybe that's helpful, maybe it's not. But at what point do we stop? Do we stop with cystic fibrosis and HIV? Or what determines what is a disease and what is normal, or what should be fixed and what shouldn't be fixed? We use genetically modified organisms to make tons of pharmaceutical products. So just knowing the sequence and the structure of a specific protein can be helpful in identifying molecules that combat certain cancers. So we can combat cancers by blocking some of these functions of these proteins. So some drugs like imatinib is going to inhibit leukemia by causing problems with the function of that protein. So leukemia is when you have too many white blood cells, and that is a type of blood cancer. And by taking this drug, it's going to inhibit the production of those white blood cells and help the person not have leukemia. Another is the production of pharmaceutical products. So things like human growth hormone and insulin are made by using things like restriction enzymes. So what we can do is take this gene for insulin, put it into bacterial cell cultures, allow those bacterial cells to just reproduce, and then the bacterial cells will secrete those insulin. So anything that is a protein that can be made from a gene and kind of secreted from a cell can be done this way. And so what we would do is then just collect the insulin from the bacterial culture, overprice it in a pharmacy, and then sell it to diabetics. Another type of pharmaceutical product is called a transgenic animal. So this is kind of going to produce farm animals, PHARM, farm, quote unquote, where what we do is we insert a gene for a human protein into the animal and allow them to produce that for us. So for example, we can see this goat has been given a transgene for antithrombin, which is a protein that prevents blood clots. And so what happens is when the goat produces milk, the animal's milk is going to contain the antithrombin, and we can use the milk that's produced by the animal to collect this protein and then again package it and sell it to people who have clotting disorders. There's many practical uses besides just medical uses for biotechnology. So we use biotechnology to compare forensic evidence as well as generate what's called a genetic profile. Some people would refer to this as your DNA fingerprint, but forensic biologists think that the term genetic profile is much more accurate. So this can be obtained by any analysis of tissue or body fluids. As long as it's got a couple of cells in it, you can usually use PCR to amplify the DNA enough that DNA testing can be done. So what you can do is you can test the DNA at the crime scene with the DNA of your suspects to accurately determine which individual is guilty. One that is a really useful method of creating genetic profiles is to look at something called a short tandem repeat, or STRs. These are really unique to specific individuals. So these would be markers of repeated units of either 2 to 5 nucleotide sequences, and then those are repeated anywhere from 5 to 35 times. And the number of times it's repeated is extremely variable from one person to the next. So the likelihood that 2 people are going to randomly have the same short tandem repeats, it's basically nothing. In 1984, there was a man named Earl Washington, who is pictured here, who was convicted and sentenced to death for the rape and murder of a girl, Rebecca Williams. He eventually got taken off of death row because there was concern about the evidence. And then eventually STR analysis done by something called the Innocence Project, which was, this is a group of people who were taking DNA evidence in 2000s and applying them to older cases when the DNA technology was not around to show either that people were innocent or guilty. And it found by his STR markers, so this is the semen on the victim, matched perfectly with a different suspect. So this man spent 17 years in prison, was finally released in 2001 after the STR short tandem repeats showed that he was actually, in fact, innocent the entire time. We can also use biotechnology in our environment. There are many microorganisms that have their natural abilities to transform chemicals, so we can exploit this by taking those genes that allow them for those transformations and putting them into bacteria. So we have bacteria that can actually extract heavy metals out of the environment and incorporate them into metals that can actually be recovered. And so this is a way of kind of getting toxic things out of lakes and rivers and other environments and removing them. DNA technology is huge for agricultural productivity. Creating genetically modified organisms are mostly plants, but we have the ability to take specific genes that speed up the selective breeding process. So instead of having to spend many generations breeding for the specific traits we want, we can take traits like delayed ripening, resistance to spoiling, resistance to disease, resistance to drought, and just insert them into the crop plant right now and have that desirable trait already. Being able to genetically modify or change the DNA of many organisms raises a lot of safety concerns as well as ethical concerns. There are benefits of genetic engineering, and they have to be weighed against harm of creating harmful products. And a lot of the harm that we think of is not something you would think of beforehand, or stuff that happens incidentally later on that you're like, oh, I didn't see that coming. The United States has many guidelines in place to help make sure that none of these hazards or harmful products get produced. All genetically modified labs have safety procedures and precautions that have to be followed. Anytime we use microorganisms, they are often crippled so that they could not survive outside of the lab, so that if we're using a virus or a bacteria and it got out of the lab, it wouldn't actually do any harm, it wouldn't be able to survive. And many dangerous experiments or experiments that are considered too dangerous are just straight out banned and not allowed to be done. There's a lot of fear in the public concern, mostly around possible hazards of genetically modified organisms that are specifically used as food. So we have passed laws in the last decade that require genetically modified organisms to be marked as genetically modified. Unfortunately, about 80% of the crops in the US are already genetically modified, so most corn, soy, and canola are already GM. So even though there's a lot of concern about this, most of the food that you eat is already been genetically modified. Some of the fear around genetically modified organisms comes from misunderstanding what it is. It's really just a change in the DNA, which doesn't necessarily inherently cause any changes that could be transferred to consumers. One of the biggest fears is something called transgene escape. And this is the idea that genes for like resistance to pesticides would get out through pollen and then get into like weed plants to create things like superweeds that would then not be able to be killed easily. This has not been a huge issue. It has happened a little bit, but most of the plant studies use mechanisms for preventing this kind of thing so that pollen is actually never being released from the genetically modified plants.